Welcome, everyone. Thank you for joining us for the Chabner Colloquium MAP series. My name is Dr. Aditya Bardia from Massachusetts General Hospital, and I will serve as the moderator for today's session, which will include presentations by Dr. Gad Getz and Dr. Keith Flaherty. Before we begin, I would like to start by thanking Novartis and Regeneron for providing educational grant support for this series. I also want to acknowledge Dr. Bruce Chabner, MAP Series co-chair, and Massachusetts General Hospital for collaborating with me and STO on development of this series. We are especially grateful to the presenters for their participation. Additionally, I encourage you to visit our exhibit hall, which you can access on the upper right corner of your screen. And you can learn more about each of our generous supporters, Novartis and Regeneron. They have posted informational brochures and videos for you to download and view at your discretion. In addition, you may set up chat with a representative if you desire. Information was emailed to attendees who indicated that they would like to receive continuing educational credit for this event. Upon full participation and completion of the pre-post-test process for each presentation, your CME certificate will be available to you. So now, without further ado, I would like to introduce Dr. Getz presenting on pan-cancer analysis of whole genomes, and Dr. Flaherty, who will discuss MAP kinase-targeted therapeutics. Immediately following the presentation, we will host a Q&A. If you would like to submit a question to be addressed, please use the chat function of this platform to post your question. Questions can be submitted at any time during the presentation. Please note that the microphones for all attendees are automatically muted. So without any further ado and comments, let's proceed with today's session.
I want to thank the organizers for giving me the opportunity to tell you about pan-cancer analysis of whole genomes, particularly Bruce Chabner and Aditya Bardia and the Society for Translational Oncology. Here are my disclosures. The learning objectives of this session is to identify non-coding drivers in cancer, power to detect mutations in drivers, and recognize the ratio of coding to non-coding drivers. From projects like the Cancer Genome Atlas, we've learned that most known drivers at that point were in protein coding genes. For example, genes like p53, RB1, NF1, MYC, PIK3CA, etc., where a mutation could cause an aberrant function of a protein or disrupt either activating or deactivating the function. And this is important because targeted therapy typically are addressed towards these mutated drivers, and that's the basis of precision medicine. There are a few drivers also detected over the past few years in non-coding parts of the genome. The most well-known is the TERT promoter mutations, but there are other ones like FOXA1 and NOTCH1, and others that have different types of functions that activate genes by changing the regulation. Keep in mind that the regulatory elements tend to be more tissue-specific than protein-coding genes. So the big question we wanted to address are there other drivers in non-coding regions of the genome? To do that and answer many other projects, the International Cancer Genome Consortium, the ICGC, together with TCGA, joined forces and created a project called PCOG, the Pan-Cancer Analysis of Whole Genomes. It's a project that recently ended. It was more than 5 years with together, the two big projects gave more than 2,800 tumor normal whole genomes of cancer across 30 tumor types, and included many researchers throughout the world and 17 thematic working groups. All the work was published quite recently in 25 publications in Nature Publishing Group, and there's a whole thread in the Nature website related to these papers. and you're welcome to visit the website and see the different papers. Today, I want to tell you about a story, one of those papers that was related to discovery of point mutation drivers in non-coding, which is part of the paper that I'm showing here. And this work was, the main players are Esther and Reiber, a former postdoc in my lab that now has her own lab at MGH, and Morten Nielsen from Jakob Skoo Peterson's lab, and Federico Abascal from Inigo Martin Conera lab. So we looked at across all these data, whole genomes, and first asked, where are mutations in the genome pile up in individual sites, individual bases? And what we could see that the most mutated sites in the genome, the most mutated site is in KRAS. The next one is the promoter mutations of TERT. But the third one is another KRAS hotspot, and BRAF and PIK3CA, etc. So as you can see from the top mutated sites throughout the entire genome, a large fraction of them are actually coding genes. But what are the other ones that are non-coding? We looked at them more carefully, and we saw that actually a large number of them are caused by AID, somatic hypermutations at immunoglobulin genes. And the signature of those mutations, the type of mutations, match the ones expected from an AID process. Another set of hotspots are associated with promoters of genes and are caused by UV, and they are in the melanoma or cancer type. You see the column for melanoma. And those were reported recently as well, that when transcription factors bind to the DNA, they essentially prevent from repairing of the UV damage, thus having higher rate of mutations. So these mutations are actually not drivers, but just an increased mutation rate due to transcription factor binding to the DNA. Third type of hotspots were caused by another mechanism, which is apobec mutagenesis that caused preferentially at edges of hairpins. And those are these ones that are highlighted here in the screen. So what we've learned from this is that coding mutations are drivers are common. Non-coding mutations are, many of them are, the top ones at least, are caused by these different mutational processes that don't necessarily mean that these are actually drivers. They could be just technical reason for more mutable sites in the genome. So given that, we actually looked for elements that are drivers in non-coding parts of the genome. So how are we finding drivers in general? We have to define elements or regions of the genome and look for enrichment of mutations or a pattern of mutations that is unexpected by chance. So we need to define those elements. Then we need to define the cohorts of patients that we're analyzing. Then we need to develop a model for the background mutation rate. So how many mutations we expect to see and what is their pattern in those elements. And then develop a statistical test that says, is the pattern that we're seeing significantly different from what we expect by chance? And then we calculate the p-value for every one of these elements. And then we need to correct for the multiple hypothesis, and we get a q-value. And those elements that have a q-value less than 0.1, we call them significantly mutated elements, and they are candidate drivers. So we define the elements as 11 different types of elements, coding genes, but also different regulatory elements like enhancers, promoters, 5' UTR, 3' UTR, link RNAs, microRNAs, etc. And then we collected all of these elements and tested for these patterns of different mutations. So here are the cohorts. This is the cohorts of the PCOG project. We had 27 large enough cohorts that you could see here, individual cohort types. And then we also aggregated them based on their tumor type, for example, different subtypes of breast cancer together or brain tumors together to CNS, all the carcinomas, etc. And also we had a pan-cancer group that represents all of our cancers together. Then we encountered a problem that we actually had 13 different methods developed by different groups to find drivers. But we wanted to leverage all the benefits of all of them. However, they are not independent because some of them use the same type of signature. For example, the mutation burden or enrichment in functional sites. So we had to combine the significance of all these methods with a new tool, and that tool is called the Brown's p-value combination method. It extends the Fisher method to integrate p-values of different tests that are correlated. So we had first to calculate the correlation structure between the different methods, and then integrate them to get a new significance level based on all the tests together. So we did that, and then we got an initial list of drivers, but then had to apply different filters based on the number of mutations in patients that we see, but most importantly, based on the sequence properties and the signatures, like we've seen in the beginning, if it was AID driven or UV driven or these kind of APOBEC driven signatures, we had to remove those sites because these are potentially artifacts, and then left out with a smaller group of candidate drivers that we believe. So what did we see when we did that? We found 705 hits in 179 different elements. 602 of them were in 143 different coding genes, and another 100 were in 36 different non-coding genes. So you could see the vast majority of the drivers that we see are actually coding genes in the genome. So we looked carefully at those 36 significant non-coding elements. And even after looking at them, we saw that there's some potential artifacts. So first of all, we were happy to see that one of the most significant hits and across many cancer types is the third promoter mutation. Well, that was our positive control, and we indeed found it. But then when we looked more carefully, we saw that 10 of them were actually potential artifacts, and 5 of them were due to new targeted mutational processes. So here I'll give you some examples. First, P53 5' UTR mutation. So we found in the UTR of P53 6 different indels that were seen to be functional. And the way we know that is that we look at the expression level of P53 across the different cohorts. Whenever it was mutated, you see the red dot has low expression relative to other samples in the same cohort, suggesting that these indels indeed change in the regulation of P53 and decrease its expression. Another gene is TOB1, and you could see that in TOB1 3' UTR, we saw an enrichment of single nucleotide variations, SNVs, and which didn't correspond to microRNA binding sites. And as we saw for P53, those that have the mutation typically had a lower level of expression compared to wild type. It's interesting because TOB1 is a transducer of ERB2, and it was significant in carcinoma and in the pan-cancer cohorts. And what it does, it regulates other mRNAs by binding to their 3' UTRs and promoting deadenylation. And when we looked at the TCGA data, we saw that actually TOB1 and its neighboring gene were amplified in cancer, in a focal amplification in that region, giving orthogonal evidence that that dysregulation of this gene is important. Then we looked at albumin, which had a large number of indels. You see these green dots there. And albumin is a highly expressed gene in liver. And what was interesting, though, that we saw that these indels not only appeared in the gene itself and its introns, but also 1 kilobases downstream of the gene, which suggests to us that these are not driver events. These are some sort of a process that generates those indels in highly expressed genes. And when we look at the other candidate drivers that we had, for example, NF-kappa-BIZ and TOB1, they didn't have this increase in this 1 kb downstream, but another FOXA1 in prostate cancer indels also showed increase in the downstream of the gene, suggesting that it's also an artifact. Then we looked at 2 long non-coding RNAs, NEAT1 and MALAT1, which also had a large number of indels, and they are also very highly expressed link RNAs. And we suspected that it is also caused by this mutational process. And we indeed saw that the indels, not only that there's an increase in indels, they actually have a different type of indels. They're typically longer indels, those green bars there in albumin, NIT1, and MALAT1, which is very different from what we see in the other parts of the genome. So that's a novel mechanism of indels that generates those indels in highly expressed genes, and those might not be actual drivers. So why are there so few non-coding drivers? So first of all, maybe we looked at all these regulatory elements that we know about. Maybe we missed some drivers in areas of the genome that are not part of these annotated regulatory elements. So we added another approach where we looked at windows of 2 kb throughout the genome and looked for significance. And here again, we found the significant coding regions and some of the non-coding regions that we found previously, but we didn't find anything new when we did this kind of unbiased 2 kb window search. Then we thought, maybe we're just not finding drivers because we don't find mutations in these regulatory elements. So we looked at the average coverage and therefore the average detection sensitivity at different types of elements. And as you can see, this cumulative probability, all but the promoters have a very good coverage that is very similar to the coding regions. The promoters have a lower coverage and less detectability. So it could be that we are actually missing drivers in promoter regions. And indeed, even TERT, the most famous promoter hotspots, we see that some cohorts have a low detection sensitivity, like the CNS astrocytoma or the liver cancers had very low detection sensitivity because of lack of coverage in this high GC region of the genome, this promoter of TERT. So we could actually, based on these sensitivity estimates, we could estimate how many TERT promoter mutations we are missing in our data. And we could estimate we're missing on the order of 216 TERT promoter mutations in this data set. These are the red bars here. And indeed, in liver cancer, we are missing many of them. And in another study that did a deep sequencing of the promoter of TERT, the rate of around 60% was found, which is the same rate that we're seeing here after we added those missing, estimated missing TERT promoters. Finally, maybe we're just underpowered. So the power to find mutations depends on the size of the cohort and the background mutation rate. So the larger the cohort, we can find drivers at fewer patients or smaller percent of patients. But if we have a small cohort that has a higher background rate, then we can't find drivers that affect a smaller fraction of patients. So as you could see on the right, the bladder cancer was a small cohort with only 23 samples, which has a moderate background mutation rate. And this is the gray bar on top. And there, we would not be able to find drivers that are 20% or less. It only could find 6 out of the 23 as drivers. It has to be at least 6 patients out of the 23. And all these yellow tumor types, we can only find drivers that are between above 10%, but below 10%, we won't be able to find drivers. So the bottom line here is that we need much larger cohorts to actually find the drivers, because the majority of drivers in cancer are much less than the 20 or 10%. They are typically, we could find many drivers below 10% and even below 5% of the population of patients. Finally, we wanted to estimate in a different way what is the ratio of the non-coding to coding drivers. So the rationale here, we said that if we look at known cancer genes, those are expected to have also non-coding drivers in them, because we know that there are known players in cancer. So we looked at their promoter regions of 5' UTR and 3' UTR and estimated how many mutations we would expect to see in those genes as a group, and how many do we actually observe. And the observed minus expected is how many we are actually potentially driver events. And And as we can see, in the coding regions, the CDS, we found a large fraction, a large number of excess of mutation. These are caused by the drivers. And in the promoters, we saw a bit of an excess, which a big chunk of that excess is actually the third promoter mutation. And in other 5' UTR and 3' UTR, it's a small number that could be even the error bar contains 0. So if we aggregate all these non-coding regions compared to the coding regions, we see a 10-to-1 ratio between coding drivers and non-coding drivers. So to summarize, what I showed you is that we described a novel and robust approach to find drivers in cancer in whole genome data based on point mutations and single nucleotide variations and indels. And interestingly, unlike disease-associated germline variants that are found by GWAS studies, most cancer drivers are actually located in protein coding genes. Not all of them, but most of them. Coding mutations outnumber the non-coding mutation, somatic mutations, as we showed. And the reason for it could be that cancer both needs large effect size events, meaning mutations that have a big effect on the function of the genes, and also can sustain large effect sizes, where the germline likely cannot sustain large effect sizes, whereas the cancer cells can, because they don't affect the entire body and don't go from a fertilized egg all the way to a full adult. Non-coding mutations are likely tissue specific, and therefore we need to increase the cohorts of individual tumor types to find the driver, because in many of them we couldn't find the frequencies of drivers that we expect. So we need much larger data sets of whole genomes. And today, still the whole exome data sets are much larger than whole genomes. So we expect to find many more non-coding drivers when the number of genomes will reach the same number that we have exomes today. But still the ratio we predict to be roughly in a 10-to-1 ratio of events. Finally, I want to acknowledge the many people involved in this project, particularly Esther, Morton, and Federico, and Inigo and Jacob, but also the entire drivers and functional interpretation group of the PCOG project, and the discovery of driver element subgroup, and the entire PCOG network, and the patients, of course, that contributed their samples, and the funding agencies. I'll be happy to take questions.
Hello, my name is Keith Flaherty. I'm a medical oncologist at the Massachusetts General Hospital Cancer Center. I'd like to thank Drs. Bardia and Chabner for the invitation to participate in this series. Look forward to taking questions after my talk, and hope that this talk, in following Dr. Getz's, helps to sort of lay out some of the content, pathway by pathway, target by target, that this series of talks will ultimately explore. I've been focusing on the MAP kinase pathway for my whole career, just about 20 years now, and I'm going to focus mostly on developments in the past several years, but hope to hit some of the foundational aspects in terms of why the field is so focused on kinase pathway, some of which were already highlighted in Dr. Getz's talk in terms of overall cancer genomics and where the MAP kinase pathway sits in terms of the frequency with which genetic aberrations affect this pathway. So just briefly, in terms of disclosures, I've been involved with several companies, as you can see here, as an advisor. There's just one that I've put in bold here that is developing a drug in the MAP kinase pathway. It's therefore a high degree of relationship that is in this content area. Having said that, I will not be talking about any of the agents they have in development, including the one in the MAP kinase pathway. So I'll avoid that entirely. The other companies highlighted here, for which I serve as a director or on the scientific advisory board, do not have agents in the MAP kinase pathway. However, a couple of the firms for which I've served as a consultant do. So just please keep that in mind. So the MAP kinase pathway, of course, is one of the best studied of the RAS effector pathways. And in the early 2000s, a fairly systematic analysis was done looking for components of the MAP kinase pathway that might harbor genetic aberrations. So RAS, of course, you're well aware of, and I'm not going to focus on RAS because while the MAP kinase pathway is immediately downstream of it, RAS as an oncogene and attempts to target it, of course, is its own entire discussion. Rather, my focus is really at the level of the RAF isoforms, BRAF, CRAF, and the lesser studied ARAF, and then MEK and ERK, and downstream programs from those mediators within the MAP kinase pathway. We don't really find an appreciable frequency of mutations in other components of the MAP kinase pathway other than BRAF. So most notably CRAF and ARAF very rarely harbor mutations. And MEK1, in any case, harbors mutations in a small fraction of cancers that don't really seem to be particularly important drivers. ERK is rarely, if ever, genetically altered in cancer. So really, BRAF is the focus in terms of somatic genetic alterations in cancer. And you can see that they're quite common. Initially described in a Nature paper in 2002, they were estimated at the time to comprise about 7% of all cancers with some form of a BRAF mutation. But I want to highlight a couple of points here that are important for the rest of my talk. One is that point mutations in the kinase domain of BRAF, specifically the V600 position, this valine that resides in the middle of the kinase domain, that is a very focal hotspot where substitutions to a handful of different amino acids can occur, and glutamine being the most common, or the so-called V600E mutations. So that's that very tall peak that you see in the middle of the kinase domain. And these are, again, by far the most common of the BRAF alterations. That said, there are other mutations that can affect the kinase domain. And you see those represented there to the left of V600 and surrounding V600. These occur in exon 11 in the case of the left-hand set, and exon 15 where V600 itself resides. And so we used to classify these mutations as simply non-V600 BRAF mutations. I'm not going to go into great detail today in terms of the biochemistry of these various mutations, although a lot of published evidence now supports the notion that these mutations actually need to be further subclassified. The V600s are treated essentially by themselves as a group, and therapeutically, that's where we've made the most advance, and I'll be focusing the next several slides on that topic. That said, these non-V600 mutations, we currently break down into so-called class 2 and class 3 BRAF mutations. And that relates to the degree to which they can drive signaling in the MAP kinase pathway, basically without any upstream cooperation from RAS. That's the case for the class 2 mutations, versus some participation, some facilitation provided by RAS. And those are the so-called class 3 mutations. We are still yet to realize effective therapy broadly targeting those subpopulations. I'm going to touch on that a little bit later in my talk. By far, the greatest advance have been made pertinent to the V600 mutation subset. And there's real biochemistry and chemistry reasons as to why that's the case. The drugs that have been effective at controlling V600 BRAF are unable to effectively inhibit class 2 or class 3 BRAF mutations. For that, we believe we need totally distinct chemical matter, and there are programs in development currently which are still at a relatively early stage. So in V600 BRAF mutant melanoma, that's where we've witnessed the most significant clinical impact, and where we really have begun to understand the determinants of response and resistance to BRAF inhibitor monotherapy, as well as the more recently established BRAF-MEK combination strategy. I'm using this one slide just to highlight the point that after the initial discovery of compounds that could inhibit V600 BRAF, and somewhat uniquely, as I said in the last slide, dabrafenib is one such drug. There are now 3 drugs in this class that are FDA approved, including vemurafenib and encorafenib. And this is a typical spectrum of responses seen with dabrafenib in BRAF V600 mutant melanoma. This constitutes about 45% of the melanoma population. About 4% of all cancers harbor these V600 BRAF mutations, and another 3% roughly harbor class 2 or class 3 BRAF mutations. What we learned, which I'll summarize in the next slide, in terms of what it is that constitutes resistance to BRAF inhibitor monotherapy, is that further inhibition of the MAP kinase pathway was supported or suggested in translational evidence as being potentially helpful. And that's now been well validated clinically. You can see here from this 2012 data, evidence to suggest that the addition of a MEK inhibitor just downstream of BRAF to a BRAF inhibitor significantly improves initial response to therapy, increases the complete response rate, and the overall fraction of patients who have objective responses. And the underpinnings of this are highlighted here. So here's the depiction of the MAP kinase pathway situated just below RAS. In this case, I'm highlighting NRAS because that's the isoform of RAS that's most relevant in melanoma. So CRAF, MEK, and ERK are highlighted as the central components here of the MAP kinase pathway. But what we learned after exposing BRAF mutant melanomas, experimentally in the laboratory, but more importantly in the clinic, with one of the now approved BRAF inhibitors, is that we see a variety of alterations that occur in signal transduction, some of which are associated with somatic genetic changes that arise. All of them, though, collectively described here on the left-hand side of the slide, reactivate the MAP kinase pathway. Interestingly, we don't see resistance mutations in BRAF itself, a common theme in other oncogene targeted therapy contexts. Rather, we see bypass where mechanisms allow CRAF to become the dominant signaling isoform, even with continued exposure to a BRAF inhibitor. That said, there are BRAF alterations. Shown here are splicing alterations, that's numbered number 3 here, and BRAF amplification that can also play a role in resistance. But specifically, we don't see the types of quote-unquote gatekeeper mutations that block drug binding that are seen in other oncogene targeted therapy stories. Notably, bypass through the PI3 kinase pathway has been described in melanoma, and we think increasingly that's an important feature of what determines resistance to BRAF-MEK combination therapy. But another way of depicting the improvement in outcomes that's been seen with BRAF-MEK combination therapy over BRAF monotherapy is reflected here, taking a few randomized phase 2 and 3 trials that investigated BRAF-MEK combination versus BRAF inhibitor monotherapy. What you see depicted here, in fact, are the actual results of clinical trials, but now analyzed as a composite. BRAF inhibitor monotherapy improving overall survival compared to historically standard dacarbazine in metastatic melanoma patients, and then the further improvement in overall survival with the addition of a MEK inhibitor to a BRAF inhibitor in this population. So clear advance led to FDA approval now for 3 of the BRAF inhibitors in combination with a MEK inhibitor. So I mentioned the BRAF inhibitor backbones a moment ago. Each of them now paired with a MEK inhibitor have shown fairly consistent improvements, such as are described here in this overview slide. Now, the story gets very interesting when you go away from melanoma and investigate other tumor types that harbor these alterations. Non-small cell lung cancer is another cancer type in which these mutations are found. About 2% of non-small cell lung cancers harbor V600 BRAF mutations, and another 2% the class 2 and class 3 BRAF mutations. So in the V600 population, what you see here with a combination of dabrafenib and trametinib is an outcome that looks quite similar to that seen in melanoma. So I won't go into great detail in terms of summarizing the data otherwise than to say it suggests that BRAF plays a similar role and serves as a similar therapeutic vulnerability in non-small cell lung cancer versus melanoma. And this is a notable subpopulation given how large the non-small cell lung cancer population is overall. However, in thyroid cancer, where about 40% of patients harbor BRAF alterations, BRAF inhibitor monotherapy has a much more muted response. That's shown on the left-hand slide with that waterfall plot. Notice that there are objective responses, about 40% of patients versus 60% responding to BRAF inhibitor monotherapy in melanoma and non-small cell lung cancer. But the depth of response is really quite muted in comparison to what's seen in melanoma and non-small cell. Interestingly, in the most aggressive form of thyroid cancer, anaplastic thyroid cancer, where BRAF mutations are particularly common, BRAF inhibitor monotherapy, and as shown here, the combination therapy, dabrafenib, trametinib, has notable efficacy and has led to FDA approval of the doublet in this subtype of thyroid cancer. But there's something going on here in terms of the molecular underpinnings of thyroid cancer, not necessarily somatic genetic alterations that would explain this resistance, but something that relates to this cell of origin or this lineage, which several groups have been working on, trying to uncover how to overcome the apparent resistance that's seen, particularly evident here with BRAF inhibitor monotherapy. Colorectal cancer is the real outlier in terms of very muted efficacy with even BRAF-MEK combination therapy. You see here a cohort of patients treated, all of whom had V600 mutations. BRAF inhibitor monotherapy produced essentially no objective responses and only a 12% objective response rate to the doublet. And this really began to define this heterogeneity of response profile for oncogene targeted therapy for the field. We've now seen other examples of this as we've more broadly explored oncogene targeted therapy across the various cancer types that harbor a common mutation. But in melanoma versus colorectal cancer is where this story really became disparate at a very early point in the clinical development of these agents. Now, there's been a substantial amount of translational work done. Highlighting here the BRAF inhibitor monotherapy result. I'm going to come back to that in just a moment, but before I do, there has subsequently now been a broader investigation of BRAF-MEK combination therapy across cancer types that harbor V600 BRAF mutations. That's what's shown here. And this cohort was included in the so-called NCI MATCH trial, for which I served as one of the principal investigators. And we specifically excluded patients with melanoma, colorectal cancer, and thyroid cancer because we already knew of efficacy in melanoma and thyroid and lack of efficacy in colorectal cancer. We were interested in understanding the broader relevance of this regimen beyond those tumor types. Now, at the time that we launched this trial, the non-small cell lung cancer data was not yet in hand, and so we did include lung cancer patients, and you can see a few of them reflected there with the red bars. But more importantly, we saw responses in other tumor types, and a fairly broad spectrum of tumor types were reflected in this cohort. And we think this is important in terms of really trying to understand this homogeneity versus heterogeneity of response across tumors as defined by their cell of origin when you're targeting a common genetic alteration. So now we're beginning to get a much more complete picture in terms of the spectrum of sensitivity or resistance to this therapeutic modality by tumor type. In colorectal cancer, what's quite interesting is the translational data here from tumor specimens suggesting that even BRAF-MEK combination therapy is not able to inhibit the MAP kinase pathway very well. And so what's shown on the right-hand side of the slide, I'll draw your attention, is the percent change from baseline in ERK phosphorylation, this downstream component of the pathway. The combination therapy has really modest effects compared to BRAF inhibitor monotherapy in melanoma, which is what's depicted on the right-hand side of that slide, where there is heterogeneity, but most patients have 80% or more inhibition of the pathway by this measure, whereas in colorectal cancer, even the doublet therapy does not achieve that degree of pathway control. So there appears to be some other mechanism that allows the MAP kinase pathway to persist in terms of signal transduction, even in the face of the doublet. And translational evidence has been developed now that pretty clearly points to a feedback mechanism, which I'll just quickly build here, that basically allows the MAP kinase pathway to be sustained through the epidermal growth factor receptor, and uniquely so in colorectal cancer compared to other tumor types. So what's shown here basically is that after inhibition of either BRAF itself or BRAF in combination with MEK, you see feedback upregulation of the epidermal growth factor receptor and its activation. That's enough to actually maintain MAP kinase pathway signaling to a degree, as I showed you on the previous slide, is known to occur in patient tumor samples. But here in experimental systems, this evidence has now, I think, become very convincing that this is in fact the mechanism. If you drive overexpression of EGFR or silence it through drug or genetic manipulation methods, you can clearly functionally demonstrate that it is in fact EGFR that's driving this biology. And so this is a unique program that's not based on somatic genetic alterations in EGFR or other components of the MAP kinase pathway, but rather is what we would say is an epigenetic phenomenon, where signal transduction and the feedback loop described here seems to mediate resistance. And so this evidence was used to launch a few clinical trials. I'm summarizing two of them here, where EGFR antibodies, which of course have been standard treatments for colorectal cancer for several years now, were explored in combination with two different BRAF/MEK regimens. So here's panitumumab in combination with dabrafenib or trametinib, or cetuximab with encorafenib and binimetinib. And now you start to see responses that look a bit more like what we see in other cancer types, but still not quite. So this is a triplet drug regimen in both cases, where response rates are still not on par with what we see in melanoma and non-small cell lung cancer with even BRAF inhibitor monotherapy. So while this biology appears to be real, supported by experimental data and now clinical data, there's still something that keeps these tumors from responding to this therapy, and we have not completely accounted for what that biology is. I just want to briefly mention in transition from the V600 BRAF population to the other BRAF mutant populations, that in melanoma at least, we've been able to extend the benefit described in patients with metastatic disease to those patients with high-risk resected disease or the adjuvant setting. And in that setting, we can see a curative effect of adjuvant BRAF/MEK therapy in patients with resected stage 3 disease. What I'm showing here is the relapse-free survival impact of 1 year of adjuvant therapy. So 1 year of adjuvant treatment, you can see here ending then at month 12 on this slide. Patients then discontinued treatment and were followed thereafter. And you can see this persistent separation in the curves where a fraction of patients are not having relapses after a few more years follow-up, suggesting, in fact, that adjuvant BRAF/MEK can eradicate microscopic residual disease in a significant subpopulation of patients. So this is the greatest degree of impact we've been able to demonstrate with this approach. Certainly, there remains room for improvement in either the adjuvant or metastatic setting in any of the tumor types in which we've investigated this regimen, including melanoma. But this evidence certainly gives us a reason to believe that this is a foundation or a pillar upon which we can continue to build. Overall survival is improved in the adjuvant setting as described here. This was the first interim analysis describing this result, not the final overall survival for which we're still waiting. So in transition from V600 to non-V600 BRAF mutations, I just want to mention that what was uncovered in colorectal cancer in terms of this epigenetic dysregulation of signaling that allows the epidermal growth factor receptor to become upregulated and mediate resistance seems to have analogies in other cancer types. And here I'm focusing on melanoma data, where we and others have now described that tumors that have relative resistance to BRAF inhibitor-based therapy, they have an epigenetic state that is different than those that are sensitive. And specifically, what's described here is that tumors that are relatively resistant have low expression of the melanocyte lineage determining factor, MITF, one of the transcription factors that controls the differentiation of neural crest cells into melanocytes. These cells have neural crest markers, and we refer to them now as being relatively dedifferentiated and neural crest-like in comparison to the more sensitive MITF high population. There are other markers that correlate with MITF and others that anti-correlate, and so one can derive signatures that describe these relatively sensitive versus relatively resistant cell populations that are readily found in melanoma patients, as described in the right-hand figure, where we've described the differential outcome on BRAF/MEK combination therapy, where patients who have high MITF and low AXL expression, receptor tyrosine kinase, those patients do by far the best in terms of outcome with BRAF/MEK combination therapy versus these less differentiated MITF low neural crest-like cell types. And so this might be a general rule that as we try to understand homogeneity and heterogeneity of sensitivity and resistance across tumor types when focusing on a genetic alteration that's in common that we're targeting therapeutically, likely there are epigenetic mechanisms that are unique to certain lineages or cells of origin that we're going to have to account for in terms of trying to understand resistance and ultimately to develop rational combination therapies in the years ahead. So now moving over to other concepts in terms of development of MAP kinase pathway targeted therapy, I want to take a little deviation towards the MEK and ERK inhibitor data that's been generated. And although I'm focusing here on BRAF mutant cancers, I do want to just pull this NRAS mutant melanoma data into my comments because it's highly relevant for our efforts to try to control the MAP kinase pathway in tumors where we currently lack an effective BRAF targeting strategy. So MEK inhibitors have been in clinical development now for about 20 years. And one MEK inhibitor, binimetinib, was brought forward in a phase 3 trial in the NRAS mutant melanoma population. Turns out that NRAS mutant melanomas are more sensitive in preclinical models than KRAS mutant cancers to MEK inhibitor monotherapy. And that was some of the evidence that motivated the initial clinical investigation, phase 1/2, and then ultimately this phase 3 clinical trial. And what this phase 3 trial showed was a modest improvement compared to historically standard chemotherapy with MEK inhibitor monotherapy in this NRAS mutant population. There's a population of patients who do respond to therapy. I'll come to that in the next slide. And those responders are the patients who have this longer duration of disease control depicted in the blue curve, that roughly 15% population who are progression-free at about 9 to 12 months. Unfortunately, most patients don't achieve that degree of benefit. They have some degree of disease control, superior to chemotherapy, but again, only modest. So there's a subpopulation, it would appear, where the MAP kinase pathway is particularly important in these NRAS mutant tumors. But I'm highlighting here just that MEK inhibitor monotherapy does have activity separate from what it can do in combination with a BRAF inhibitor in the V600 BRAF mutant context. So the objective response rate in this trial with a MEK inhibitor monotherapy was 15%. So I just want to highlight that number. This has become a sort of an anchor approach to try to target tumors that harbor BRAF mutations, that particularly the class 2 and class 3 BRAF mutations, while we're still developing direct BRAF targeting strategies for this population. We think a MEK inhibitor is certainly a well-qualified approach, room for improvement, no doubt, but does have some evidence of efficacy. Unfortunately, this trial, which was primarily directed towards showing a progression-free survival difference, showed no impact in the secondary endpoint of overall survival, ultimately did not lead to FDA approval. And so just summarizing these results to say that MEK inhibitor monotherapy clearly does have activity in tumors that are dependent on the MAP kinase pathway. Apologies, this slide came out of place here, so let me just skip ahead here. So we're really interested in the prospect of developing MEK and now ERK inhibitors as well in tumors that have, again, these so-called non-V600 BRAF mutations. So class 2 and class 3 BRAF mutations, as I described them before. And this is some of the first evidence that supports an ERK inhibitor specifically in such a population. You see here color-coded patients by tumor type and then also by genotype, if you look at the bottom portion of this slide, clearly showing activity. And I would say it's actually quite comparable to what we saw with MEK inhibitor monotherapy in RAS mutant cancer. I not included in today's talk data suggesting a roughly 15% response rate for a MEK inhibitor as monotherapy in the BRAF V600 melanoma population also. So fairly consistent signal in terms of the degree to which these drugs can produce responses, but unfortunately in only a minority of patients. Nonetheless, we think it's a foundation. Another ERK inhibitor beyond the one that I just described on the previous slide, BVD523, has intriguing properties and we think might actually be one upon which further clinical investigation could be built. This is an orally available inhibitor that has a very slow dissociation constant. That's what's being described here with the residence time figure at the bottom of the slide. And because of this slow dissociation constant, this is a drug that, although it's an orally available drug, we felt could be dosed in an alternative schedule that might increase the therapeutic index, and most notably a weekly schedule as opposed to a daily schedule, where rash and diarrhea become limiting. We thought this might be intriguing to explore an alternative schedule. And we and colleagues explored this in a phase 1/2 trial. You can see that on a daily dosing schedule, we could only safely administer 40 milligrams daily, whereas on a weekly schedule, we could administer a much higher dose, 250 milligrams. And this type of pulsatile strategy with a drug that has such a slow off rate or dissociation, we thought actually might offer a really unique profile. When we look at the efficacy data, it's still quite preliminary. With this weekly schedule, you can see a dose escalation cohort is depicted here. So patients received a variety of different doses. These patients, of course, many of them were heavily pretreated. Nonetheless, some promising early data suggesting that there's clearly some efficacy with this weekly schedule, and this is currently being further explored. As I mentioned briefly before, we've been awaiting the development of RAF inhibitors that can directly target class 2 and class 3 BRAF mutations. And one such drug is shown here, LXH254. This remains an investigational agent. It has been explored to some degree clinically as monotherapy in a phase 1 study, but more clinical investigation has been done in combination, and here shown with an ERK inhibitor. So a two-drug approach in the MAP kinase pathway, somewhat analogous to the BRAF-MEK combination approach in BRAF V600 cancers. And an intriguing early signal has been demonstrated here in a mixture of patients by tumor type and genotype. But this is a group of patients who had non-small cell lung cancer, the vast majority of which had KRAS mutations, which again are not entirely equivalent to class 2 and class 3 BRAF mutations, but still establish some proof of concept that this next generation RAF inhibitor approach might be worthy of further investigation in those BRAF subpopulations. So we now have this next generation coming along. Their potential role as monotherapy and in combination with MEK and ERK inhibitors is of significant interest in the field for all of the subtypes that have class 2 and class 3 BRAF mutations. So I'll conclude by saying that we've really made substantial therapeutic inroads for V600 BRAF mutant cancers, and that really is the group for which I would say we have an adequate formulary of therapies now with several regimens FDA approved. There's still room for improvement, though, and whether some of that improvement could be gained by further improvements targeting intrinsic components of the MAP kinase pathway, or whether we now need to shift our emphasis to other mechanisms that drive resistance, some of which I've alluded to in my talk today, that's an area of ongoing translational research. It's in the other BRAF mutant subpopulations, class 2 and class 3 BRAF mutations, where we have to put the most emphasis right now, because while we've shown evidence of efficacy for MEK inhibitor monotherapy and ERK inhibitor monotherapy in a subpopulation of those tumors, it's only 15% or so that do respond. We're really anticipating the rush of new RAF inhibitors. There are now several moving forward, and we're very hopeful that those agents might constitute the next pillar of treatment for those important subpopulations of MAP kinase pathway activated tumors. Thank you so much for your attention.
That was excellent. Thank you so much, Dr. Getz and Flaherty. Excellent talk and great launch to the series, starting with the genomic landscape and therapeutic relevance. So thank you again. We have a few questions from the audience. I'll start with the first one. This is to Dr. Getz. There is evidence of pathogenic super enhancers in some cancers. What do you think of lack of enhancers showing up in your genome-wide analysis?
Thank you. Thank you for the question, Aditya. There are several reasons for us not seeing a large number of these super enhancers in our dataset. One is that some of these super enhancers are affected by translocations. And that was the second part that I didn't talk about in this paper, about looking for structural variations that bring different parts of the genome next to other genes, and by kind of what's called enhancer hijacking of one gene to another gene. And that was not what we discussed, because we talked about point mutations. There's another section of that story about looking for driver structural variation. So that's one. The other thing is that we looked for one type of mutations actually in the binding site in those enhancers that are across many different parts of the genome, like CTCF binding sites and things like that. And those we didn't consider all the types of enhancers that bound by the same transcription factor across the genome as one element. That's another analysis that we could do in the future. And the third reason, which I mentioned in the talk, is the power. We just didn't have sufficient numbers of patients in our study to find them. So we could find them, individual events, but were not frequent enough to find to reach significance. So that again, as I mentioned at the beginning, those drivers are typically tissue specific. And for each, although we had the largest ever collection of whole genomes, for every particular tissue type, the numbers were not that great, so the power was not sufficient. So what we need is to really reach the numbers of, like we have for whole exomes, which is in the numbers of tens of thousands of exomes, essentially one order of magnitude larger in the number of genomes. And I wish we could get something to 100,000, so that would give us sufficient power to find all these types of drivers. But additionally, there's definition of the regions, as I said, and looking at structural variations, etc.
Thank you. Another question. This is for Dr. Flaherty. You nicely mentioned the heterogeneity in response to BRAF inhibitors and BRAF/MEK based on tissue of origin. Is there some role for BRAF signaling to promote immune resistance? And could the fact that MSS colorectal is often poorly immune responsive account for worse outcome to BRAF inhibitors?
It's a great question and an area of very active research. So I guess you can take it from either direction, which is MAP kinase pathway activation. Does it suppress immune recognition or even the ability of infiltrating lymphocytes to attack tumor cells? Is one way of thinking of it. And we and others have done experimental work, but also translational studies and analysis of patient specimens before and after short-term exposure to BRAF inhibitor-based therapy. And we've seen evidence, both in models and in patients, that inhibiting the MAP kinase pathway, particularly in the setting of a BRAF mutation, does result in increased infiltration of T cells and activation of T cells. That doesn't prove that that's the direct mechanistic consequence, but it would suggest a relationship there, that there is essentially, as the question is posed, kind of an immunosuppressive effect, if you will, of MAP kinase pathway activation. That's been suggested in other oncogene contexts, by the way. It's not unique to the MAP kinase pathway, but it's an interesting related point about MSI high colon cancers, which have a large mutation burden, somewhat responsive to immunotherapy, but they commonly also harbor BRAF mutations and aren't particularly responsive to BRAF inhibitor-based therapy. And I would say there that the through line really extends from the work that I summarized, suggesting a very different signaling process in colorectal cancers, whereby sort of bypass of BRAF blockade is almost instantaneously achieved through this alleviation of feedback and receptor tyrosine kinase signaling. Still for reasons that we don't totally understand. We think that is the biochemistry that undermines or underlies resistance. But why is it that cell of origin lends itself to that? We still really haven't quite worked out.
A lot more needs to be done, which is great. A lot of exciting translational work, as you highlighted, related to this field, and you've led the way with development of these drugs. Another question, and this relates to passenger versus driver mutations. A central tenet of precision medicine is genomic analysis, and this distinction of passenger versus driver. To both of you, could you comment on, from a genomic perspective, can over time some of the passenger mutations become driver because the way it's analyzed is based on frequency? And could the presence of passenger mutations impact the function of driver mutations? And from a therapy perspective, again, do the presence of certain passenger mutations impact the response? So coexisting mutations with BRAF impact the response to BRAF inhibitors.
Keith, why don't I start and then you continue for the BRAF?
Yeah.
The vast majority of mutations are passenger events in the genome. Very few are driver events. However, there's, as the question implied, there's some passenger events that are actually drivers that we just don't know yet that they are drivers because they are belonging to genes that are so rarely mutated that we will need more patients to find that they are drivers. So these are subset of the passengers, but still the vast majority of passengers will remain passenger events. Now, in terms of a clinical implication or function in the cancer, they do have implications because they reflect the signatures or the processes of generating mutations in the cancer, those passenger events. And as Keith mentioned, the MSI, or you could have a BRCA positive patients that have their own signatures, like the homologous repair defects or mismatch repair deficiency, etc. Those are reflected in the patterns of passenger mutations, but represent real vulnerabilities of the cancer, which could be exploited with PARP inhibitors and other kind of new development of drugs against MSI-related vulnerabilities, etc. So they could be kind of a signal or a pattern that reflects response. Now, in terms of resistance, passenger mutations could be totally passengers in cells, meaning they didn't give any fitness advantage to the cells throughout their history of the cells, but they could become drivers of resistance under therapy. So they could be a pre-existing mutation that could not have given any benefit before. Once you give a drug, could become now a driver under that treatment, and that is you could switch a mutation from being truly a passenger to becoming a driver, depending on the context of the cells and the treatment. Keith, to you.
Yeah, I would just quickly add. I mean, there's a third term that we commonly use, which is subclonal driver, which I think the question is also kind of implying, right? So a true oncogene that has true biologic function in terms of contributing to at least the subpopulation of cells in the tumor becoming cancers in the first place. And actually, in melanoma and BRAF resistance, the easiest one to point to would be NRAS. So in a BRAF mutant melanoma, we essentially never see coexistence of an NRAS mutation in the untreated state. So patients who just have surgery for their melanomas. Yet in the setting of BRAF inhibitor monotherapy, 20% of the time we saw emergence of an NRAS mutant subpopulation that also harbored a BRAF V600 alteration. Right, so both alterations, and extensive work was done thereafter to try to understand is this a pre-existing subclone versus a truly, if you will, de novo point mutation in NRAS, and all the evidence that's been generated suggests that it is a pre-existing small clonal subpopulation that has relative lack of fitness. So in other words, having both a BRAF and NRAS mutation turn out in experimental systems to induce oncogene-induced senescence, more powerfully so than either NRAS or BRAF. So while they can occur, if you will, in nature under the ultraviolet radiation-induced mutations, cells that pick up those two mutations have relative lack of fitness. When you inhibit BRAF, you're effectively making that cell behave like an NRAS mutant cell that lacks a BRAF mutation. You're sort of neutralizing the BRAF component. Then it gains fitness. But in experimental models, in those same cells, you then pull away the BRAF inhibitor, they again lose fitness. When I say lose fitness, they don't die, but they don't grow as well. And so that's there are many other examples of subclonal drivers that have been implicated in therapeutic resistance in other settings. But that's our best example in melanoma and BRAF, I should say more generally.
Wow. And that's fascinating in how the patterns of passenger mutations impacting driver mutations and how things can change over time. Again, emphasizing a lot more work needs to be done. Thanks for clarifying and emphasizing this. So to wrap up, I would again thank the presenters, Dr. Getz and Dr. Flaherty, for an excellent launch to this series. Our educational grant supporters, Novartis and Regeneron. Our exhibitors, AstraZeneca, Foundation Medicine, Genentech, Novartis, Pfizer, and Takeda. Now I would hand this over to Dr. Chapner.
Aditya, thank you very much for moderating this session. I find it just fascinating how the growing complexity of genetics is explaining what we're seeing clinically. And that is really the theme of this series, the translation of science into effective therapies for cancer patients. Thanks to Gaddy and to Keith for brilliant talks, and to our sponsors, Novartis, Regeneron, and Sanofi, and also to the staff of STL for organizing this. I've learned a lot from this. I learned that I don't understand cancer as well as I thought, and that there's so much more to learn about the genetics, epigenetics, and the function of non-coding portions of the genome, which influence the growth of cancer. I look forward to the following 5 sessions. I hope you'll all join us for that. Thank you.